I said they must come in. Well, I think that meeting doesn't have a password or something. It's kind of walk around
I remember that I didn't scan today.
No, I think we must just bring the scanner.
But they don't move the chair.
So we have to fit in. What are you telling me?
Don't put the chairs in. Touch the
Good afternoon, everybody. Good afternoon, everybody. We're just waiting a few more minutes. We're running 5-10 minutes late. Just bear with us while we fix some challenges. There's also a session next door. There's quite a number of individuals, medical officers who want to join the session. So we are going to check with them. No, we're not starting now. Let me go and get the emails. All phones. Good afternoon, everybody. My name is and I have the honor of facilitating this session. I'll start by introducing my co-facilitator. Do I call you Charlotte? Okay. My co-facilitator is Dr. Charlotte. We were talking just a few minutes ago in one of the other sessions, talking about this doctor story that's confusing. So one of my, most anxious periods in my life was when I first got a PhD. So I go to church, or used to go to church every Sunday with my mom, mainly because my mom insisted that if I don't, I won't get into heaven. But here's the problem. So my mom, could not stop talking about this clever, intelligent son of hers, who is now a doctor, but did not explain what type of a doctor he is. Now, as things go with church, Catholic Church, a lot of the people who go to church are old ladies whose health is failing mainly. And my biggest fear was some old lady fainting and them saying, doctor, doctor, doctor. I contemplated stopping to go to church, until my mom could explain that this doctor cannot help you. You faint at your own risk, So, Dr. Maseremola, I understand why you don't want to be. She has a PhD in molecular medicine, which means that she can't do anything for me if I faint now. And I'm joined as panelist by Dr. Janine Schofield, who also, I suspect, is not useful when it comes to matters of fainting. However, I'm quite happy that I see some medical officers, so we're safe, right, guys? Some clinicians are in the house, so you can faint all you want. They're going to help you. So Janine has a PhD in human genetics from UCT, postdoc at University of Oxford, Norfolk medical fellow. And her interest, when she gives the opening remarks, she will give us a taste of what her interests are. Joined on the panel is Veron. I was going to say Professor Veron. He's a Sante senior researcher at the University of KwaZulu-Natal. Of course, we had to include UK terrain. We are in their city, and Zulus are notorious for not being friendly with people who ignore them. So he represents the city here in today's session, and he works on the area of metagenomics. By way of explanation, this session is a series that we started sometime last year. So we conduct these webinars with global leaders in clinical research. The areas that we discuss include everything from regulatory to strategic research agenda and others. It's a series that is sponsored by Amgen, one of the biggest biotechnology companies in the world. And I think if you've been following some of the sessions this week, you will realize that clinical trials diversity is an important matter, and I'm hoping that they will give us some insights as to why it is so important. I think it's important to a point where a lot of regulatory authorities are requesting that developers include a statement on how they mean to achieve the diversity that's required. Pharmacogenomics is an area of interest. Drugs don't work the same way in different genetic profiles. So it's also about innovation and creating effective therapies. But I'm not an expert in that, I'm just the leap. You can tell that I come from a long line of priests. So I'm going to hand over to them to introduce themselves, and then once they've done that, we will then open the session for discussion.
Okay.
She's the co-facilitator.
Right, so I'm Janine Schofield. I'm a research group leader at the Council for Scientific and Industrial Research, the CSIR. My group is called the Bioengineering and Integrated Genomics Group, BIG. My team needed some morale uplifting during COVID, so that's what we renamed ourselves. But it covers all manner of sins. We primarily focus on making stem cells from individuals of African descent. So it's an induced pluripotent stem cell technology, not embryonic. But with those, we can make just about any cell type in the body. So we primarily make liver in a dish, liver organoids in a dish. And alluding to Dr. Nsomi's comments about clinical trials, we're really trying to create innovative tools like these liver in a dish cells with which we can sprinkle, so to speak, drugs and try and understand how individuals of different genetic background metabolize or break down these drugs, so that essentially we can get to a point where we can mitigate the 1 in 12 people being admitted to hospital for adverse drug reactions in South Africa.
All right, thank you. So I'm Professor Veron Ramsaran from the University of KwaZulu-Natal. I'm a research professor. I have a background in human genetics. And we focus specifically looking at infectious diseases. And we have, in KZN, we have, some of the worst problems with HIV, TB, sexually transmitted infections. So this is what we look at in this area. And, recently we've also looked at COVID as well, and we find that there is a vast amount of genetic diversity that exists within each of these diseases. And what we find is exactly the pharmacogenomics play a major role. We've found some new publications where we show that the polymorphisms identified in the Western countries don't follow through within African countries. And we find that we have to look at new genes, new genetics within this population to identify what are the factors that are contributing towards the effects that we see within this environment. So I guess this is a good place to kickstart why diversity is important within clinical trials.
Thank you so much. So you touched on pharmacogenomics, and I want to get to immunogenomics in a moment, but let's not make assumptions about everybody understanding what diversity means. So shall we get into some definitions, please? So what does diversity mean to you in a clinical setting?
Well, that is a very good question, because there's many forms of diversity. There's ethnic diversity, there's gender diversity, there's age diversity. We can go on. There's many different aspects of diversity. And the more diversity that you can have within a clinical trial, the greater the chance of success of that drug or product going into market. So we have to be inclusive in terms of all of those different aspects: gender, age, ethnic, race. All of that has to be included in any clinical trial. And traditionally, what's been going on is that a lot of the drugs that come into the market in Africa have not been tried in Africa first. I'll give you a good example within the infectious disease space, HIV. We had this massive Thai trial that was going on with HIV, and it worked beautifully in that environment. They brought the vaccine to South Africa, failed miserably, because it was not tested in an African market. The entire genetic makeup is completely different. So it tells you that vaccines and trials can work wonderfully in different countries, but when you bring it into the place where it needs to work, it won't. So diversity is important. Do you want to add on?
I had this read thing. So you answered. I just want to, but to add in perhaps some examples that add to it, and I think particularly for this audience of our next generation of medical practitioners who are really at the coalface of what you're going to see when it comes to this. We've known traditionally that for a lot of the clinical trials, because they were primarily done on white males, okay? That's adults, white males. So we've just done a study with some adverse drug reaction information, line by line listing every single report that's been published in the last 5 years, 25,000 of them. They came through reactions with various reports within them. And we see the same thing that everyone else is seeing around the world. The female to male ratio is 2 to 1. Twice as many of those reactions happen in women compared to men. I'm not going to go into the details and the stats, but age then becomes an issue as well. So we're literally seeing problems that have existed, That's why a lot of people advocate, and Linda Gale Becker did it beautifully last year as well, for diversity in clinical trials.
Thank you for those opening remarks. And I think I'm just going to add a chairperson's prerogative. You would notice that BioAfrica is located in Africa. So we prioritize that, and I think the panelists have indicated the role that genetics play in the effectiveness of these therapeutics or vaccines or other medical products. One of the biggest challenges is, or rather one of the biggest agendas that we're grappling with is how to develop local innovation so that we set our agendas. I'll give you one of the most fascinating anecdotes or a story that I experienced. We went to a cancer research meeting where a professor or researcher from Nottingham showed a slide on the distribution of cancers around the world. Because there are a few, obviously there's a lot of environmental factors, but as well as genetics. The second slide was what research is being done where. So you have the prevalence of certain cancers, high prevalence in the global north, geographical global north, breast cancer, and a whole lot of those. And then in the global south, we have our own unique cancers that are more prevalent. In this part of the world, because of the diet and stuff like that, you will tend to find a high incidence of esophageal cancer and a lot of colon cancers. And part of it is caused by the toxins in our staple foods, the way we store them. However, when they showed the slide of what research is being done in the global south, we were doing research for the cancers up there. Nothing for our cancers. And that's a political agenda that is driven by money, but it's also something that we're able, or we could be able to overcome if we get to define our own research agendas, starting from recognizing what the problem is. And I think with that, I would hand over to my fellow chair, co-chair.
Thank you so much. So we've spotted the problem, and that it's complex, and we want to prioritize ourselves as Africans. Who defines the agenda? Who sets the framework for what we should focus on? How do we manage that process? And what does advocacy in that area look like?
Yeah, I think that's also a rather good question. I mean, I asked this question in one of the sessions I was in yesterday as well, is that, you know, a lot of what is happening right now in terms of genomics and genetics as well as clinical trials is defined by individuals or individual companies or individual entities. I think there needs to be some sort of government involvement in this. There needs to be some sort of coordination by government so that this actually works in a certain way. Because what's going to happen is that each entity is pulling towards their own agenda, and there's no coordinated effort where all of this is happening. You know, there's a lot of genomic trials that are happening right now, and one of the biggest problems is that of all the human genomes that have been sequenced in the world so far, only 3% come from Africa, which is shocking. We have the greatest genetic diversity in Africa. I mean, just take for an example, Madagascar alone is just an island, small island compared to the rest of the world, has more genetic diversity in Madagascar than the entire Europe combined. And that shows you the amount of diversity within Africa. That's just looking at Madagascar. Imagine across Madagascar to all of the other African countries. Now that tells us, and we only sequence 3% of all the human genome sequenced thus far, only 3% come from African descent. How are we designing clinical trials? How are we designing vaccines? How are we designing any of these things when we don't know what's going on? So I think, in my opinion, that the individual governments have to drive this agenda forward. They have to identify that this is a problem, and we have to know what drugs and what vaccines are going to be given to the individuals. Once that is set up, individual companies and entities can then move that agenda forward.
Can I ask a question, Janine? Before you do human clinical trials, presumably you do preclinical work.
Yeah.
One of the things that we noticed during COVID was how the paradigm was upended. So if you think about HIV, which you spoke about, 40 something years later, there's not a single effective vaccine. But COVID happened, we had vaccines galore. It was like being in a jumble sale of vaccines. the way they were being developed. But I want to just use, Janine, the work that you currently doing at the CSIR. Is there an opportunity in terms of innovative tools for us to leapfrog some of these of the backlogs, that instead of going into human trials, you could change the paradigm a bit and repackage how we do clinical development?
We need hours. Yes. So I'm trying to make sure it isn't an avalanche of thoughts and it's clear and concise, and this is a topic that's very dear to me. So in a traditional clinical trial pipeline, for example, prior to getting to first in human, there are preclinical tools that are used, and essentially they are increasingly complex variations on models, right? So you might have in silico analysis of the drug, there might be some biological understanding of the vaccine. It's going to shift to then and this is where our area comes in, cellular models. So modeling what impact, how this drug might be to cancer cell line in a dish. These are all in a dish, we call them in vitro. Subsequent to that, and each time you do this, you have to reduce the scale of the experiment that you're doing, right? Into the tools that you're doing. So you can start with hundreds of thousands, and then you get to animal trials, and you've really got to hone down your candidate drugs by that point, and then you get to your phase 1. What is the stat? Is it, I think 75% of drugs fail phase?
More than 95.
90, sorry, thank you, and making my point in a minute. So one of the things that we're trying to do, and actually, funny enough, this did actually happen with COVID, with some of the vaccine technologies as well, is that cellular assays, if your cells are really, does anyone know the story of Henrietta Lacks? Healer? Healer cells, good. You should all read the book. If you haven't read the book, you need to go and read the book. So I'm not gonna pound on those cells, but those have been the workhorse cells that contribute to the polio vaccine, 2 Nobel Prizes, etc. But from a genomic integrity point of view, having been grown in a lab for 70 years, they're not genetically stable. What we are increasingly trying to do, and what the FDA has recently recognized, is that, and animal trials are expensive, okay? They're still important, and they're not going to stop. They're still important because it's physiological. What we are trying to do is go, what synthetic biology, and I'm talking genome engineering, the stem cell technologies that we do in our lab, what can we do to make those HeLa cells more relevant, more physiologically accurate, so that we can reduce that timeline of, I can turn to Dr. Rose Hayashi at NWU and she can go, thanks, with that information I can reduce the animal experiments I have to do down to 3 instead of 100, as an example. And that's where we're trying to push that increased understanding. And the thing that the world has been missing for so long is African genetic diversity in these cell models. You said 3% whole genome sequencing? It's 3% of global induced pluripotent stem cells that we make are of, and I say this, African origin. Not one has been sent into the European repository that was made here yet. And we're trying to change that.
Wow.
Well, so I have, like Dr. Tommy mentioned, so I have my PhD in molecular medicine and molecular immunity. So, and part of my PhD project showed us that there's genetic contribution to host susceptibility to disease. So that's why I wanted to get into immunogenomics. So pharmacogenomics, awesome, but I have a problem that I need you and your team then to solve. So we have a data problem, right? So we have identified that, say, there's certain mutations that are in Far Eastern populations that make them more immune to TB infection. But we don't have enough data in the African population. And that requires for me to say, get a clinical cohort so get research clearance. These are my human subjects, but I will have to draw blood, all of that. Is it possible for you to create such a repository?
Can I start?
Yeah, sure.
Because I think you're going to,
Yeah.
I was going to ask for a really extraordinary infectious disease. So I'm going to add for my limited infectious disease. I make models. That's what we do. But I came home from the UK after my postdoc, and I was hired because I was asked to create stem cells from cohorts of African individuals who had been clinically curated as elite controllers. So we know that, to the best of my knowledge, the CCR5 null mutation doesn't exist. Yeah, but we have elite controllers. We know that there are people who are refractive to HIV infection here. We know that some of it's HLA, and there are few, and you're going to speak maybe more to that, but this is exactly what these models can do. We're actually working on a project with the NICD at the moment where they've identified a novel mutation that appears to make their cohort less susceptible to sepsis. That's a big thing, right? And it's very broad. How do you create a model that answers the question? Because you've got hundreds of people that you're looking at. So the data, you need lots of people to try and isolate the causative nature of the mutation that's resulting in that. If you take a skin biopsy and turn it into a stem cell, which is what we do in a dish, and you use genome engineering, and you just make one simple, that mutation, you just create it. Now the only difference between the two lines that you've got, the two stem cell lines that you can turn into blood cells in a dish, and we do, the only difference is the mutation you've added. You can determine what the contribution of that causative mutation is. And that's where the power of African genetics comes in, because, like Corinne was saying, those mutations are unique and sometimes rare in European populations, but prevalent here. So that's where those tools can come to first.
All right, great. Thank you so much, Janine. So I think, one thing that we have is a lot of information already, because we have centers that have collecting samples for ages. I mean, we've got Caprisa, we've got ARI, we've got essentially there's a lot of different places. All we have to start doing is now collaborate across different centers. Yes. And that's what we do really badly in Africa, is that we don't collaborate with each other. Yes. What we do is we work in silos and we try to cure the disease ourselves. But, I mean, samples collected for HIV work for TB, work for cancers, work for everything. I mean, it's one human, one sample can be used for every single disease. Now, I think if we start creating almost so that was my previous comment, was that government coordinates everything. Because, for example, if I champion it, excuse me, Janine. Janine is not going to collaborate with me because, it's Veron. So that's what happens. To be honest, scientists have a lot of egos. But if it's coming from like a government type of sector where we have the central data bank or central biobank, that's what everybody's now pulling into a resource. That's the way that needs to go forward. We have, believe it or not, several million samples stored in repositories across South Africa. But now we want to go and collect more samples because we don't have enough. We have enough, we're just not using it well. And I think with the limited resources that we have in Africa, we should tap into what already exists and use this to answer questions. I mean, we do have information. We've published some work where we've shown that there are some genetic mutations that are associated with TB, with some of the drugs, where they are rapidly metabolized and slow metabolized, and so on. So, there is data that does exist. So what also happens, and I'm sure some of these guys in the audience will realize as well, is that when you come in with a PhD, you do 200 samples. You collect it for 1.5 years, and then it's stored in your supervisor's repository, and it's forgotten about. But those 200 samples add up when you start looking at everybody in this room that starts collecting 200 samples. So I think we need a more coordinated effort, basically.
Can I just come in there because you've touched quite the raw nerve. What you're advocating for borders on unethical, because when you collect the sample, you obviously have to get the consent. And post your project, you have to reapply for that consent. One, to me, is when I shoot you.
Yes.
This is the CEO of our regulatory authority. But there is an element and there is an issue of ethics involved there. One of the biggest fears now, one of the biggest discussion points is the fear that we are going for another Berlin conference. Do anyone of you know what the Berlin conference was? So the powers in Europe in 1885 met in Berlin and decided to carve up Africa. And they allocated themselves different countries. The Belgians took Congo because they had diamonds. Others took gold and all of that. And the fear, and part of the reason why this clinical trials dialogue is happening is that those samples are the new gold. If you mine them and you know how to mine them, you are going to generate massive wealth. But I think you want to answer that.
Yeah. No, exactly. So, this is something that we've also been thinking about, because when you consent samples, you obviously have to consent for future, thinking in the future. So, maybe the existing samples may not fall within this, but the way that Caprisa has done it, the way that Ari's done it, has done it so that they're looking at genomics, and the samples can be stored and the samples can be used for future research. All the documentation kind of states those things for some of the projects. But I'm saying for all new projects going forward, we now need to think of ways that when we invest money in collecting a clinical trial or research, we need to think about how is it going to benefit Janine's work? How is it going to benefit other people's work down the line? So we're not wasting resources on collecting individual cohorts around South Africa. How can we pool? Also, there's another issue about ethnic diversity. Coming back where, how does individuals of Zulu descent differ from individuals of Xhosa descent, differ from all other ethnic groups? South Africa is very diverse, and it comes back to the question that you mentioned earlier, what is diversity? And how do we know? I mean, South Africa is very diverse in the sense that we have a lot of different ethnic backgrounds. Is that going to affect our clinical trials in the future? And I'm sure it is. So we have to, now if you do a clinical trial in KZN, will it work in Eastern Cape? So,
Janine wants to report.
So this is such a great session. I've got 2 points, and they're kind of 2 points that have been made. So I'll go to your last comment now. So, do you guys remember at some point there was a frightful moment where chloroquine was going to be used? Yeah. If you look at the Xhosa population, the percentage of a genetic variant in the G6PD gene, which comes up in red blood cells and is responsible for metabolizing chloroquine, is, I think the percentage, it's present at 0.8%. It's minor, it's not a problem. In South Africa, in the Tsepedi population, it's present at 16%. That's one country. So this is what Aaron was talking about. So we do need to take that into consideration, and I think trying to create those definitions, ethno-linguistic often comes up and is helpful, but those are things that we need to think about. So I don't have the answer for that, but I think we need to think about it a lot. Going back, and I'm going to be careful about the way I say this, but leading to your point, about ethnicity and samples being the new trade, the gold, the resources, the diamonds of Africa. I think we need to recognize that this country and this continent harbors resources that are hugely valuable, and we need to not hold on to them ourselves, because I want to work on these things, and I know you do, and we all do, but if I'm the only person working on it, it's going to take me 100 years to come up with a new drug. If I enforce global access, 10 drugs will come up in 10 years. But we need to be careful about just giving this away without local IP and local resources, whether it's financial or skills improvement or whatnot coming together. And so one of the things that we've been thinking about in terms of moving away from these silos is, and for example, our group isn't, we don't want to duplicate the amazing genomics work that's going on. H3 Africa is trying to make a big difference. There are many groups who are doing that. So we're starting to think about digibiobanks in terms of if we can get clinically curated, let's take African liver disease. There's a particular autoimmune hepatitis. And those phenotypes don't exist in Europe. But we have unique data sets here, but they need them because they need to apply that to the increasingly diverse populations. And if we could create access where that data is stored here, but we create differential cost structures where pharma pays, and they pay big to access, but local academics and people in that country can access at cost, I think we can maximize those resources and at the same time make sure that drugs that come back here have been tested on our diversity.
Excellent. So we've done the preclinical work. We've got the 10 drugs, and we're getting into clinical trials. Given the hesitancy that we've seen with vaccination in South Africa, for example, how do we then get people to participate so that we in practice, in clinical practice, then we can see the impact of these wonderful solutions that we've developed?
I'll answer. Do you want to go first?
Yeah. How do you always ask good questions? No, that's a wonderful question. And it's true. It's true that South Africa is very hesitant about vaccination. We've seen this with COVID. We've seen this, amongst many different platforms. And this is something that we have to seriously start thinking about, how to basically increase vaccination, how to, and I think clinical trials in Africa will help with that. I think if people understand what is happening, because right now, a vaccine is developed in the US, in the UK, in another environment, and simply said, guys, take this vaccine. Why? How? What? It doesn't mean anything, right? So there's no education that comes with it. So when a vaccine is developed in South Africa, there's slightly more trust that comes in with it as well. There's a lot more, kind of camaraderie that comes with it as well. So now people start believing that this vaccine works for them, as opposed to a vaccine that is just developed somewhere else and just pushed down our throats. Excuse my language. But in that environment, we're now thinking about our own people in our country that we can now design. We're creating jobs, we're basically doing clinical trials, and we're doing that. So it's education that comes with it as well.
I think educate something that, and also as scientists we don't talk enough about is community engagement.
Yes.
It's so simple. Like this should be like a baseline for every ethical submission for research. It has to happen. If you want the trust of the communities, then you should go and work and make sure you have that. So it's not my area, but it's something that we've instituted in the projects where we're starting to secure sample access. That has to be a baseline. It's just scientific dissemination. If you cannot explain and get the trust of the community that you intend to serve, I don't see how we can really responsibly say that we are serving the community. So that's one thing. The second thing I would say in terms of a broader aspect of to get trust, and Vern, you kind of alluded to this in terms of government, and Prof. Naked spoke about this yesterday as well. I think there's an opportunity for a pan-African response. So if every single country in Africa is trying to create their rules for accepting this drug, a rule for accepting this one of it, and there are regional priorities. And obviously those priorities are also going to be regionally, genetically contributed to as well. But we can start creating pipelines that can apply to each country where the African Medicines Agency, for example, could say that here's your baseline from which to work, take ownership and implement this according to the priorities that you see fit based on what the clinical information is. And I think we need to start thinking about doing that.
Okay, thanks, Janine. We are going to take 3 questions from the floor. I'm hoping that you're sufficiently inspired and your neurons are firing. Anyone? And if you don't raise your hand, I'm going to call you.
Yes, a lot of people were nodding and saying yes.
Yeah, let's see.
We're going to start pointing.
Any question? There is Dr. Kaleza.
Yes, afternoon to the audience. Thanks for the interesting topic. I wanted to ask Janine specifically. Okay, no, I wanted to ask Janine specifically because if we are looking at the genetic diversity, especially like we are saying in Africa, the population is so diverse. I wanted to ask with your stem cells, how far are we from having in vitro cell lines that will allow us to study our diversity in Africa in a petri dish in a way to fast track the development of new products to the market? Thanks.
I think there was another hand. We'll take all three questions together. So there was another hand.
Oh, hi. Thank you so much for such a stimulating conversation. I found myself jotting down a few points, but it's been a whole interesting plethora of ideas. But just on some of the last points we touched on with regards to community engagement and how I think one of the panel members, had mentioned how fostering an environment where innovation happens in and amongst ourselves as a community in South Africa allows us to then gain trust from the greater population in terms of them accepting or better accepting our innovations. I think one of the points that unfortunately we are unable to get around and perhaps we might overlook it and it might end up coming back to bite us is the fact that within our own country, we also have our own issues amongst ourselves. So I think traditionally we still live in a society where in our country, science, and even though we're working on transformation and so forth, science is still seen as an elitist sort of thing that happens behind closed doors. And because we are so deeply intertwined with government in terms of how they regulate, authorize, and sometimes even fund these innovations, most of the population sees us through the government's eyes. So we're sort of behind the government in terms of when the population is looking at us. So some of the issues we face is that our population doesn't seem to have a very good relationship with how government, which then builds trust issues from our side as well, which is where the conspiracy theories are coming from, when we have well-intentioned projects or innovations that are meant to benefit the community, but because of those trust barriers, we're finding it difficult to penetrate through and reach the communities that we're working in. So I think that's also something we would do well to also look into.
A third question. Please don't pass the mic, Father. I was going to request a comment. She's giving me the look. And I think somebody else wanted to ask a question.
Yeah, once again, thank you for the interesting discussion. As she said, it's a lot of information. A lot of this stuff has to be processed thoroughly. But then my question is, okay, the professor pointed out that there's a need for multidisciplinary research. It's a very important need. So my question is, would it work if the government and the private sector or other sponsorship programs were to initiate specific like NRF funding or any sponsorship funding that would be dual based? Let's say it's dual amongst a chemist and a physicist, or dual with a pharmacist and a geneticist. Would it work if we were to force kids to collaborate from a young age so that they would grow with the skill of knowing that they need to research with each other, so that when they tackle a problem, they don't just tackle the problem from their own perspective, from their own faculty or faculty of learning? Yeah.
So I'm going to answer that question and then acknowledge your point of how I understand what you're saying, and then I'll be quick. Our liver cells, we are predicting are 2 years away from a pilot study. So I'm in trouble because I said that out loud and now I have to do it. So we're it's not recorded, is it? Yeah. So we're about that. Our current liver model can metabolize the drug midazolam, which sounds a bit lame, like why would you choose that? But it's the gold standard for a pathway that's responsible for metabolizing 50% of the drugs in the market. So we're there. It's still not a perfect, we still need to get the other genes, making sure that their levels are equivalent to primary human hepatocytes, for example. But so my team's very clever. So we're close to answer that question. I understand your point about trust and going back on. What I would say though is that I've been working for the CSIR for 10 years, and I work with the most extraordinary, my bosses and my directors and my colleagues are the most extraordinary individuals. One in particular, Sachaba, who works on IKS in particular, and his framework of research, which is extraordinary, is primarily centered around working with communities first. So he goes to community leaders, and that's how he's able to acquire very particular samples. So I think examples like that are the right way to follow. We are launching a Digi Biobank project, and I've been wanting to do it for 5 years. And the only reason it came together is we finally found the critical mass of the right clinician, the right ethicist, the right molecular biologist, the right chemist. And I could go on. So we've learned. You can't do geneticists, and you need a whole group of individuals with expertise. And that's why it's moving now.
All right, thank you so much. I'll start with your comment first, is that I completely agree. I think there is a lot of distrust with scientists because, traditionally it was take samples from us, do the research and not give any feedback at all. And that's going to create that distrust anyway. I think there has been some change that has happened and is starting to happen. We are, starting to engage with traditional healers because a lot of, the community still, go to traditional healers, and I think there's a lot of value within that space as well. So, even within UKZN, I'm going to be biased here, is that we do have, a center for traditional medicine as well, and this has been included. It's actually one of the well-funded centers within UKZN right now. So I think there is a lot of space for growth, but I think we're moving in the right direction with that as well. To answer your question, I think that's a brilliant comment. I think that's exactly what we want to do. To be honest, if you have a student that has spent time in different labs, for example, I was in Harvard University, and then I was in Texas, and then I was in different places. I'm slightly biased to those labs when I do research because I've come from those labs. So now you always go back to them for different aspects. So it's the same scenario here. Like, if you get a student that spends some time in a virology lab as well as a genetics lab, they will have bias towards those type of fields. So when they look at collaborations, they will always think about virology as well as the genetics. So that's a very good idea. I think we should look at these kind of dual programs where you can almost foster collaborations from a very young age to get that going. Speak to anybody that spent time overseas. When you want to have an in-depth conversation, those are your go-to people to talk to about anything. So I think brilliant suggestion, yeah.
As Janine said, we could be here the whole week.
Yeah.
We are running out of time because the next plenary starts in 5 minutes. But in closing, and she is going to close as a co-chair. No, you're going to close. But I would like to invite you to our webinars. I'm sure there will be communication from Africa Bio. As I said, we hold these webinars every second month. And a lot of the topics that are being discussed here generally tend to be focused on one webinar, because you can't cover the entire ground. It's just that we are all here today, and that's why we sound greedy in terms of information. So I think I would like to invite you to that. And looking at the audience, with the exception of a few of us, there is a session, the plenary that is starting now, it's Rising Stars. where some of our rising stars from across the continent will share their war stories in innovation. And I think in innovation is important. The multidisciplinarity that you talk about is anchored in the sense that if you're doing product innovation, innovation is not defined as a single science endeavor. It's defined as a multidisciplinary endeavor because you're making a product. You will also need humanities. The way human beings consume products, and drugs, vaccines, and the lots are products, is defined by their psychological proclivities. And so it's not just the science and the technical bit, it's also the psychological bit. In innovation, we speak of design thinking. And design thinking is really a concept that says you start with empathy. In other words, the product design and the work that you do is defined by the person that is going to be the consumer in the end, whether that's a patient or somebody who is a consumer from a consumer health perspective. And I think with that, I'd like to hand over to my co-chair, and from the bottom of my heart, thank my panelists and yourselves for the questions that you asked as well. And I would like to see you in our next series of webinars. Thank you.
Thank you so much, Dr. Msom. So I will wrap up by saying it's very clear that we need African solutions to African problems, and we are all in agreement about that. And I'm glad that we have a multidisciplinary audience in here. So we have hardcore scientists, we have clinicians, we have social scientists in the building, and other sessions that are important are the science, diplomacy, and communication workshops. So I would like to encourage all of us in our respective orbits to start engaging with the people around us in a very simple way, in a very unassuming way, answer all of their questions in order to establish that trust from just your networks, right? With that, I would like to thank you so much and thank our speakers. Diversity in clinical trials is an absolute necessity. This is something that we cannot run away from, and this is something that we will prioritize going forward. Thank you so much.
I am scared. Okay. I am scared.
Oh dear.
Hello.
All right.
That's what you're going to be doing.
So,
Next time I'm doing my details.
Yes.
This one is.
I was told I was this one like 5 minutes before.
No, more like an hour.
No, it was 15 minutes. It was 15 minutes and I had 10 minutes.
But so if maybe can be done here.
That's exactly why we need to do it here. Now, for example, you can increase age expectancy in the future because the treatment that they get in the Western world is designed for that population. So when we give that same treatment to us here, it doesn't work because we don't know the genetics profiles of those individuals. Thank you so much.
Hi, just want to introduce ourselves. We actually work at GKZN.
Okay.
Yeah, we're doing clinical trials as medical
excellent.
officers.
Oh, wonderful,
yeah,
wonderful.
yeah.
And whose department are you in?
So I was actually King Ed was the site.
Okay.
I was hired by Fitz Health.
Okay,
But I was
okay.
working under infectious disease.
Oh, really?
Yeah,
okay, with
yeah.
what's his name?
Prof. Musa.
Musa.
Yeah,
Yes,
yeah, yeah.
yes. Excellent.
No, this was a great talk, and I
Oh,
mean, it
thank
puts
you.
quite a lot of things into perspective.
Yeah.
The diversity, we always just sort of think, just different
No,
ethnicities,
it's
but it goes.
extremely important. And yeah, I mean, do reach out to me. You can.
Oh, can I take it?
details. I don't have a card on me.
Because we were also talking about the mistrust of vaccines,
It's a
even
major
amongst
problem.
healthcare
Okay, well,
workers.
that's true. I mean, to be honest, we are African first before we are researchers, before we are scientists, before our profession, right? Because I mean, you've been taught this from the time you're a child,
Yeah.
that there's a lot of distrust in the terms of space, and so I mean, healthcare workers didn't even want to take vaccines during the COVID
They were
space.
the worst, I thought.
Yeah, no, exactly. And yet, to be honest, I did this study when I was in my PhD, where I went to a certain hospital, and I went and just looking at healthy volunteer samples. Found that 50% of our nurses were HIV positive. Now,
Was that incidental findings?
that was incidental. I was looking for healthy, positive, HIV negative individuals.
This was 2000?
This was 2008. Yeah. So, and almost 70% of them said they didn't want to know what their result was afterwards.
Oh man.
Yeah. So,
How do we, because I honestly think that until we have the buy-in of all or the majority of health professionals, it's going to be very difficult then to get it out there into the community, because remember we are also part of the community, so
And
we
this
can't
is-
be giving
And this is,
misinformation.
yeah, exactly. So this
how do
is, to be honest, what happens, right? One of the, comments that we made was that you have a lot of these adverse drug reactions. What happens is that you take a pill, you get sick, and a go-go that's in a village somewhere feels that she's getting sick with this drug. She takes it,
Side effects,
but she thinks it's because of the drug. So she throws it away, goes to her traditional healer, and he's like, I can fix you, I can solve your problem, stop using the Western medicine.
messes with her liver.
It works for her. It basically, he's doing something that's protecting her. It's working. Because she stops taking the drug, right? The symptoms go away. Right, and she thinking that it's the traditional medicine that's actually helping her, right? So it works.
Yeah.
She goes and advocates for this everywhere, telling her that telling everybody, look, this Western medicine basically tried to kill me. And here these guys came and fixed me. Now, these things have been happening for generations. And it's still going to continue happening until we realize that the drugs that we're giving people are not the genetically right drugs to be giving people. And that is what the problem is simply that we just don't know genetically whether the drugs that we're giving is the right drugs to the right person.
So then the right thing is to say that upfront
Yes.
to the patient.
And to be honest, we have to say we don't know. It was not tested in a population of this
I know
decent.
one drug, like the chlorothiazide.
Yes.
I don't know if that sort of testing was done.
No,
because it's usually-
to be honest, a lot of the testing is not done in any.
So
to
how
be-
do they advocate
So
it
to be
as
honest,
first line
okay,
for-
so what they do is,
Where's-
do you know where they
for-
test it
for-
on? They test it on African Americans,
Oh,
right?
okay.
So that's exactly the point. So African Americans came from basically two sides in Africa. It was Kenya and Nigeria.
Yeah.
So that's why they were basically moved across to the US. And then now when they say they test African individuals, they test it on this very small pool of people that represents an entire continent, which I just told you, the genetic diversity in Madagascar alone is compared to the whole of Europe.
Those people are also diversified. in
Exactly,
unique
more
mix.
since then.
Yeah.
So now the problem is they are claiming that they've tested it on African individuals,
but
it's
they
not.
haven't.
It's not really.
So we haven't even tapped into
We haven't
anything.
even tapped into anything. So that's the biggest problem, is that when we bring a drug, and that's why I'm saying, I'm advocating for government for every single drug coming into this country. We need to do do a clinical trial to see if it works. Come up with all the fact sheets to say, okay, these are all the different types of clinical symptoms that come from it.
Yeah.
What do we need
Which
to do
ones?
to change the drug? Now I've got